RNAi versus CRISPR. To understand more about gene A, it is crucial that we find a way to control the genes that will alter the genome. Both RNAi and CRISPR accomplish this in similar yet different and specific ways. RNAi, or RNA interference, causes a silencing effect by using two types of RNA to complete the process, siRNA and miRNA. miRNA and siRNA bind with a protein to create an RA-induced silencing complex, or RISC for short. As this occurs, the RISC includes a single-stranded segment of RNA, which is called the guide strand. The process is specific as the guide strand will have the perfect complementary bases to some part of the mRNA of the gene that is attempting to be silenced. Once the guide strand base pairs with the mRNA, the way that the gene becomes silenced depends on the type of RNA the guide strand is. If it is a piece of siRNA that pairs to the gene, then it will degrade the mRNA. But if it's a piece of miRNA, then it will not allow the mRNA to undergo translation. This differs from CRISPR, although it is another method to control a gene and also has a specific process. CRISPR, or Clustered Regulatory Interspace Palindromic Repeats, is based on a natural process used by bacteria protecting themselves from viruses. Bacteria make a Cas9, which includes two pieces of RNA that were made when invading DNA was found. This enzyme can be used to cut DNA. It knows where to do this as one of the pieces of RNA will match the sequence of the DNA, and this is called the guide strand, just how RNAi also has a guide strand. As this is a process in bacteria, it can also be used in living cells. Cas9 will bind to an area in the nucleus of a cell called PAM. It will then unzip the DNA and the guide strand will find its match and cut there. The DNA will try to repair itself, yet this will cause mutations, making the gene not work. The specificity of this process is the fact that a specific piece of DNA can be added in and the DNA will base pair, coding for a specific trait. A major difference between CRISPR and RNAi is the fact that CRISPR can target multiple genes at the same time. In this case, I would prefer to use CRISPR. This is because a big difference between CRISPR and RNAi is the fact that CRISPR can not only turn off a gene, yet it can add a specific trait to the gene to find the exact effect of the gene. This allows the findings to be even more specific to the exact trait scientists want to isolate versus researching all of the effects a gene might have. Both techniques will achieve the same ending of understanding more about gene A, yet I believe that CRISPR method will give a more useful answer to specific traits that are important.